Okay, yeah.
Got it. Okay.
Okay, let me see. Should we start with the introduction, Mike?
Go for it.
Okay. Hello, everyone, and welcome to today's session. My name is Marta De Gia. I am a Syngap1 and a director of the Syngap Research Fund. We are very excited to continue the Syngap Research Fund webinar series. The goal of the series is to empower your communications with clinicians as you get more clear knowledge of Syngap. Our talk today is treatments in development for epilepsy syndromes, opportunities for Syngap1. I have the pleasure to introduce today's speaker, Dr. Ana Mingorance. Dr. Ana Mingorance is a neuroscientist specialized in genetic epilepsy and an independent consultant in orphan drug development for neurological diseases. She is a scientific advisor for the Draves Syndrome European Federation and is a strong advocate for patient involvement in drug discovery and development, and frequent joins forces with the patient communities to accelerate the development of new treatments. Dr. Mingorance received her PhD from the University of Barcelona in Spain and completed her postdoctoral fellowship at the University of British Columbia in Canada. A record version of this webinar will be available on the SRF website under webinars on the family menu. By the end of this presentation, you will have the opportunity to answer your questions. We love to hear from you. Please write your questions in the Q&A. For those of you just joined us, welcome. And again, our speaker is Dr. Ana Mingorance, and her talk is treatments in development for epilepsy syndromes, opportunities for Syngap1. It's now my pleasure to turn things over to Dr. Mingorance. Welcome.
Thank you, Marta. Gracias. And good morning, West Coast. Good afternoon, East Coast. Good early evening from Europe. I hope you can all see my slides. As Marta said, I'm a scientist. I work, my specialty is development of treatments for epilepsy. I work in general normal epilepsy, but for the last 10 years, I worked more in the syndromes that have epilepsy, and Syngap1 is one in this umbrella. I know all of these disorders are a lot more complex. It's not fair to call them epilepsy, but for the main topic of the presentation, I focus on that part of the disorder. And what I will want to discuss with you is what we had recently, but in Spanish, in another webinar by the Syngap Research Fund, which is in the first part of my presentation, the state of research in treatments for epilepsy, how the industry has taken care of epilepsy and how their interest has moved now towards the rare epilepsies, including Syngap1. And on a second part, we're going to look at several of the treatments that are right now in development for many of these diseases and which of that type of treatments could work for Syngap1, which of these are opportunities that could eventually make it to Syngap1 and to your children. So we start in that first part by looking at the entire field. You all know a bit how this entire field looks. There are a lot of rare syndromes that have epilepsy as part of the neurological symptoms. And not every person with these syndromes has seizures, but many do. And that's why we tend to cluster them all and talk about rare syndromes with epilepsy. You could probably see your logo on the slide. There's so many others. And the important point here is there are many rare syndromes that have epilepsy, and they all have different causes, different genes, different things that are malfunctioning in the brain that cause the syndrome. But for many, many years, we couldn't see this. We didn't know what was inside that big black box. We could only see that there were a lot of people that since they were children, they had a neurodevelopmental disorder and epilepsy. And we couldn't know that these were all individual different diseases. So the only thing we could treat from the pharmaceutical industry is the symptom that they all have in common, the thing that we can see, and that's epilepsy. So epilepsy as a field for drug development has been tremendously successful for the industry. And there are more than 25 drugs approved. I never want to give the number because I don't know now if it's 28, 29, we're near 30. And it started in the 1800s with bromide, which is still being used quite a lot in some countries like Germany, but mainly in the last 40 years, you can see that vertical increase in the number of drugs approved. A lot of this success in this field is because of a couple of reasons. One, clinical trials in epilepsy are a lot easier than for many other diseases because we have discrete events that we can count, which are seizures. And you can tell this patient has 10, 20, 30 seizures a month before treatment, and after treatment, there are this many. So that makes trials a lot easier than, for example, measuring cognitive activity in an old person with Alzheimer's. Those are very difficult. Counting seizures is easy. Also because there are many, many patients. Epilepsy as a disease affects tens of millions of people. So because it's chronic and there are so many patients, it was easy to do trials. We also are lucky that in general, when you clone these genes that are mutated and you put them in a mouse or a rat, usually they develop good epilepsy. So you can use mouse and rat models and other models to test drugs and then go into the clinic and do that in patients with the same type of epilepsy. And this has worked really well in epilepsy as opposed to other fields where we have done great advances in mice, for example, ALS, and then everything fails in the clinic. That has been very difficult. For epilepsy, it's a lot simpler. It seems to translate very well, brain to brain, mouse to man. So for all of those reasons, the epilepsy space has been very attractive for a lot of the pharma industry and the companies. And we have so many drugs, but we have many drugs, but not many types of drugs. And what I mean by that is that when we look at the mechanism of action, that's how scientists when we look at how drugs really work, there are not that many different ways. And essentially, all of those 20 plus drugs do one or four things. They either increase inhibition in the brain by acting on GABA, or they reduce excitation by acting on sodium channels, calcium channels, glutamate. That's pretty much it. So it looks like we have many, but essentially they're all doing four things to either increase inhibition or reduce activation in the brain. Now, the problem of this is that there is not so much variety of types of drugs. And as a result, a third of people that have epilepsy continue having seizures, even if they try the 27 drugs. And we know that particularly the type of epilepsy that is most difficult is the one where epilepsy is one of the symptoms, and there is also neurodevelopmental impairment associated, not just plain epilepsy. So all of these rare epilepsy syndromes that I'm talking about, we happen to be in this box, in this third that is more difficult than the rest of epilepsies to treat, because all of these drugs that were developed happen to do just a couple of things. So this made the field of research in epilepsy transition from a first stage that was very successful. It was a boom. There were so many drugs approved, and you can see the last 40 years, there's so many, and they all had a very broad label. So they were approved for focal seizures or generalized. So if you got approved, you had 30 million patients. That was huge. It made a transition to a second stage where the field was too mature. We still had a third of the patients that we couldn't treat. Nothing really worked for them, just partial reduction. It became harder and harder to recruit for trials, because imagine if you're a neurologist, you still have 17 more drugs to try on your patient before you invite them into this experimental trial. And then there was a commercial problem, because if you're the 28th drug approved for focal seizures, how many people is going to hear about your drug? So that almost made companies stop working on epilepsy. And this is recent. This is like in the last 10, 15 years, where it was so overcrowded that big pharma freaked out and left the field. So what happened then? rare diseases meet epilepsy. What happened then is that a lot of the focus on medicine, not just epilepsy, moved from what we can see outside, from the symptoms, towards the genes and being able to treat what is inside. First, we started by being able to diagnose all of those epilepsies and being able to open that black box where we didn't know that there were many types of different syndromes. Now we could see them. And if you look at the dates in this graph, you see that it's very recent, that is about 2010, so just the last 10 years, that we find most of the genes. And that's because before we could only sequence genes one at a time. So we could only find the family link epilepsies, the ones that are inherited, that you know that they run into the family, and so you could find them by looking for segregation. So if grandpa has it, grandma doesn't, you can find the difference. It's only in the last 10 years that we could do panels and sequence the whole exome or the whole genome. And thanks to that, we know that there are more than 400 genes that when mutated will produce a syndrome with epilepsy, more than 400. That is huge. And because of that granularity, because we can now separate the epilepsy into individual diseases, we just started a second boom in the field of epilepsy. Now the focus is on the rare diseases, orphan epilepsies, and this is because we're still seen as easier than other disorders. We still have seizures. You can still count seizures for trials. And also because even though there are less patients as a rare disease, you also have a lot less competition. And remember, you don't want to be the 28th drug approved for focal onset seizures, but you could be the first approved for Syngap and many of this. So that made it a lot more attractive to companies. In this boom, we've changed from talking about epilepsy in singular to talking about epilepsies in plural. And if you look at the clinical trials, how they look 10 years ago, how they look now, it is completely different. In 2010, trials were for focal onset seizures. They were known as partial and generalized for adults or pediatric. That's the whole thing you have. And the pharma companies that were working on that were the very large pharmas like GlaxoSmithKline, like Pfizer. I was working at UCB Pharma back then. And now, most of the clinical trials are for the syndromes. Lennox and Draves, some of the most popular, but you have many others. And the companies running these trials are different. These are smaller companies, often coming with different type of treatments like genetic approaches. They're trying to fix the biology. So it has completely changed. We're not talking about treating symptoms for tens of millions. We're talking about symptoms and treating the cause of thousands or less. So the first wave of companies that became interested in the syndromes were mainly companies that had normal drugs for epilepsy and wanted to avoid the overcrowded market. They didn't want to be the 28th approved. As an example, Epidiolex, so cannabidiol from GW, they chose to do a couple of syndromes, even though they could have developed their drug for epilepsy in general, because of commercial reasons. It is still a normal anti-epileptic drug. There is nothing unique to the diseases that they choose to develop it for. But they're doing it because there is a lot less competition, and yet there is enough of a market. So usually in these disorders, the first trial that we see is a drug of this type, an anti-epileptic drug, nothing special for your disorder, but the trials are for your disorder for commercial reasons. I don't want to say by this that it's like bad, because actually from the research patient groups, from the research foundation perspective, it's tremendously useful, the first trial. It makes a before and after for your disorder. Before you've had any clinical trial, usually you're at risk that companies look at your syndrome and they think, wow, there is not enough known, or this disease is not ready. Once someone runs that first trial, it completely redefines that. Now companies not only consider you ready, but they will start asking, hey, why are we not doing that disease that other company is doing? Should we be doing a treatment for Syngap1, for example? They hear about it. They know now, because of that first trial, what the regulators want you to measure, usually seizures. So that's the endpoint. So they know the endpoint they're going to get asked. They know it is possible to find enough clinical trial sites. They know it's possible to find patients. They also know how many patients regulators want. And they know that the regulators already know your disease. And that is tremendously useful because FDA cannot be up to speed with all of these rare diseases. And sometimes you go to them together with a company to run a trial, and they're not sure, the regulators, that you have a rare disease. They could tell you, oh no, Syngap1 is not a separate disease, it's maybe a subtype of Lennox-Gastaut, I don't know, or a subtype of who knows what, and they don't really let you do that trial that you wanted. So once the first one has happened and you've ticked all those boxes, now your disease will get a lot more speed in the number of trials that come and the speed they come. That first one is critical, even if it is with a boring drug. I like those first boring drugs for this reason. I also like the ones that come after, and that is the ones that are not just using genetics to diagnose a disease, but to treat the disease by going after the genetic problem. So more recently, it's not so much those companies that have normal epilepsy drugs that are coming, but we see biotech companies that have genetic approaches that allow them to go after rare diseases that are genetic, and they could do any rare disease that is genetic, and those are thousands. It's more of these companies that start getting interested in epilepsy syndromes. And why is that? Well, because as before, our clinical trials are easier than for many other genetic disorders. So even if you have a genetic treatment for one of the syndromes, and it's clear that that treatment is going to improve many of the symptoms, we also know that for the trial, the main symptom we're going to measure is seizure reduction, because it's so easy and so objective, so clear if it works. The regulators want it. And it's so much better to have seizure frequency as your endpoint, as what you measure, so much better than to have autism symptom reduction. That one's a nightmare. Seizures are a lot more straightforward. Thanks to that, we have many companies that could have gone after any neurological disease with their programs that are choosing to address the genetic problems of the rare syndromes with epilepsy, because we're easier than the ones without epilepsy. So that's a good thing of having epilepsy when it comes to developing therapies. So let's talk specifically now about Syngap1, about the treatments that all of these wonderful companies are developing for many of the other epilepsies, and which type of approaches those are. And is it something that we could think would work for Syngap1 or not? Spoiler alert, only one is the one I don't know if it will work, but for most of those, you have so many possibilities to address the dysfunction in Syngap1, so many. I've divided the type of treatments in 4 boxes. The first one in red is symptomatic, and those are the epilepsy treatments. But the other boxes are the ones that address what is not functioning well in the disease. And that could be by acting at the gene level or at the protein level with either traditional type of drugs or with antisense oligonucleotides, and we'll talk about what these are, or the gene therapies that use virus. So first class is symptomatic, and the other is 3 different type of treatments that could help us rescue the gene or the protein. So from the symptomatic side of drugs, and I told you a bit about this, we have epilepsy drugs that are developed for the syndromes, but they are just epilepsy drugs. They're not unique for these diseases. The example of Epidiolex that I mentioned before, CBD could be developed for pretty much any rare epilepsy. They chose to do it for Draves, tuberous sclerosis complex, and Lennox-Gastaut. They could have gone after Syngap1 and many others as well. It was a commercial decision. There's nothing special in the drug. They wanted to do the largest rare epilepsies, and those are these 3. Another drug has a different business priority, that's ganaxolone. The company developing ganaxolone, Marinus, has a drug that is acting by increasing GABA. And that's something that could benefit pretty much any epilepsy. But they chose to focus on epilepsy syndromes that had 0 drugs approved. They didn't want the large ones that had drugs approved, like Epidiolex. They wanted new ones. They chose CDKL5 deficiency, and they finished the trial, and it was good. And now they're doing PCDH19 epilepsy, but they could have chosen others. So this type of approach, using an epilepsy drug, but then developing it for a syndrome, is that something that could be an option for Syngap1? Well, absolutely. And there are many recent approvals or still in development that make a lot of sense biologically for Syngap. For example, well, we know Epidiolex because there is data for it, and other formulations you guys are sometimes using, but fenfluramine, which was more recently approved for Draves syndrome and other epilepsy, that is an epilepsy drug that was initially tried for patients that had, I call it here, reflex epilepsy. That's instead of spontaneous seizures, these are seizures in response to a sound, to startle, to lights. In the case of Syngap1, you have that type of seizure in response to feeding. So that is something where I could see potential for fenfluramine, for example, but also there is a drug recently approved, perampanel, and there is another in development, soticlestat. These are for other syndromes that they act by lowering glutamate in the brain. Remember, that's one of those classical mechanisms, but that is something that will make a lot of sense for Syngap1, where you have an excess of glutamate signaling. So many of this, biologically, medically, you could totally use them. It's just a matter of getting a company that decides to choose Syngap1 for them. In this second category, when I'm going to be talking about drugs, that the drug itself is a normal drug, it's like a drug you will take orally several times a day or one time a day, but that are designed to act on the protein that is causing the disease. I'm going to give you two examples, one activating and one blocking a protein. So I like this modality because you don't have to put things like injections into the brain. This is just a normal medicine, but it's acting on the protein. So it's like the best of both worlds of gene therapy and pharmacology. There is another epilepsy called KCNQ2 encephalopathy. These children have, from the two copies of their KCNQ2 gene, one is not working and one is working. This is also what happens in Syngap1. You have one bad copy, one good copy. But because their gene produces a channel that can open and close, a company called Xenon is developing a drug that is an opener. So because they only have one good gene, they produce some of this channel. It's just not enough. The opener, that is a normal medicine that opens the channel, will make sure that from those channels they have, they will achieve all of the function that they need and compensate for having one bad gene. This is doable because a channel can be open or closed. A second example, and just by chance happens to be the same company, is an inhibitor. So this is another epilepsy that you might all know, because also I know in the rare disease community, many of the rare epilepsies know families in other organizations, SCN8A. SCN8A is caused also by spontaneous mutations, but in this case, it's a gain of function. This is a sodium channel. Their sodium channels are too active, way too active. So what Xenon is developing is a normal drug. This is a normal medicine, their liquid product, but that reduces that activation. So they can have, without needing gene therapy, they could really increase activity of the channel or reduce activity of the channel and treat these disorders with a normal once a day or twice a day medicine. Is this something that we could think for Syngap1? I'm not sure about this one, because Syngap is not the protein at the end. That is not a channel that we can open and close. It's more of a protein that is present there and binding to the right other proteins, or is not there, but you can't turn it on and off so clearly. I don't know, maybe your biologists will figure out a way to do that, but this doesn't seem a very obvious approach, but all of the other approaches are going to be good for you. Another therapeutic approach that is being developed for some of these epilepsies are ASOs, antisense oligonucleotides. That's a mouthful. I like to call them a little fragment of DNA, because that's what they are. So this little sequence of DNA, they're quite famous now in the rare disease community because of all of the N-of-1 trials, these ultra rare diseases, when a scientist manages to make an ASO for one child, like Mila with Batten and some examples. So these little pieces of DNA are programmed to recognize a gene, and they are administered into the brain by infusion. You get an infusion, like with chemotherapy and things that are by infusion, into the brain of the spinal cord. They will get into neurons, find the gene, and they can be programmed to do two things, either increase expression or reduce expression. So if you have a disease that is produced because there is a buildup of a bad protein that happens in Huntington and some neurodegeneratives, then you want to produce this fragment of DNA that is going to find the gene and reduce expression because you don't want it to build up. But if you have the other type of disease, where the problem is that you don't have enough production of the protein because one of the genes doesn't work well, then you want your little piece of DNA to travel to the brain, get into the cells, find that gene, and increase expression. And you can do that from the good copy. So in Syngap1, you have 2 copies of the gene, like we always have, and one doesn't work. But from the good one, you could increase expression. These diseases where you have one good, one bad, are called haploinsufficiency. Haplo means half, so you have half of the sufficient protein. This little bit of DNA could help you have enough, not just half, of the protein that you need. Here's an example of that already going on for another syndrome with epilepsy, quite famous right now, Draves syndrome. Draves syndrome is due to a similar gene to the one I told you before. This is SCN1A, and from the 2 copies that we have, one doesn't work. But the company called Stock Therapeutics is developing an ASO, the little DNA fragment, to increase expression. And that's what you would do, like in the case of Syngap, when you need to have more because you have one of these half sufficiencies. It is right now in clinical trials, and the question is, well, can we do something like that for Syngap? Yes, this is a no-brainer, because actually the same company, Stoke, has shown in cells in the lab that you can do exactly the same to get more Syngap. And what you can see in their studies, and if you look just to the 2 graphs from the right, is those arrows that go up. Using their little DNA fragment, they can get the production of messenger RNA for Syngap up and the protein Syngap up. And that's exactly what you need to exploit the fact that there is a good copy, so that this little DNA fragment will increase expression and give you enough and get you out of your half sufficiency. Now, something interesting I want to highlight for you is that this is something that can be done not only using a small DNA fragment, an ASO. You can actually do this with other type of drugs. And the best example is what happened with spinal muscular atrophy. That's a regular drug that can do the same. So if you look at my cartoon at the top, there is a very famous drug in the rare disease community that is Spinraza. This is a drug to treat children that have spinal muscular atrophy. That's a very bad motor neuron disease, that they're so floppy that they can't move their head, they can't move at all, they can't breathe well because of all the muscles involved. And these babies were dying before they were 1 year old. So Biogen found a way to do one of those small pieces of DNA to increase the expression of the protein that they have too low expression. And they do that with an ASO, that is a DNA fragment. And that has to be through spinal infusions into the spinal cord. This is wonderful because the kids were responding so well that the regulators actually stopped the clinical trial and put everybody immediately into the treatment arm. So out of the control placebo group, everybody on treatment. That was great, but it's still tricky because you're doing infusions into the spinal cord to get into the CSF. So another company, Roche, one of the largest pharmas, found a way to do exactly the same, but with a traditional drug. And you see here the picture. This is oral solution. This is no different from all of the other drugs that your children are already taken. So this approach of designing an ASO or a normal drug that will bind to the good copy and increase expression has been done, and will be a wonderful opportunity that biologically should be doable for Syngap1 as well. And then we get to the last category. These are classical gene therapies. When we think of a gene therapy, what we're thinking is a virus. You take the DNA out of the virus, all of the virus DNA, and instead you put in a gene that you want to bring to the brain. And now you use that virus to put it into the brain. This is one injection. This is not infusions every couple of months. This is just once and done. The virus gets into your neurons. It gives you the gene that you wanted. In science, we like to use the very small virus called AAV, adeno-associated virus. You don't have to learn that, but there are also larger viruses. And what you do is to, have the gene that you need to add for your disease when it's diseases that are missing some gene. So right now there are gene therapies in development using this, a virus carrying a gene. for CDKL5, SLC6R1, and several others. They're still not approved. Some are still not even in trials. For other diseases, there are gene therapies approved, not yet for the epilepsies. But another approach that is particularly used if the gene is too large, but you still want to use the small virus, the AAV. And we want to use the small virus because it's the one that is the best at getting throughout the entire brain. Another approach is that what you put inside is not the gene, if your gene is way too large and doesn't fit, but the boss of the gene. And I know I explained that kind of weird, but it's essentially another gene that is a very small gene that once in the body, it's just going to get your gene to overexpress. It's the boss of your gene. This is called a transcription factor, and those are smaller. And this is something that is soon to start clinical trials for another epilepsy, for Draves with SCN1A. SCN1A is too large, but the company wanted to use the best virus, which is the AAV, the very small. So what they put inside is the boss for SCN1A. Is this something we could do for Syngap1? Yeah, again, absolutely. You could think of doing this straightforward gene therapy where you have, in your case, your gene is large, so a large virus like an adenovirus or a lentivirus carrying Syngap1 as a gene and get it to the brain. And that's, again, one injection. Or we could think of the approach where we use the Syngap1 boss, a transcription factor, that potentially will fit the very small virus. And then you can use the best virus to get to the brain and still achieve an increase in Syngap1. One super important message here. There are diseases where neurons don't migrate to where they should, or connections are not formed well, or there is neuronal death, and those diseases exist, and those are much more difficult to treat. But it appears from what we know in the clinic, and in particularly what we know from mice with Syngap1 deficiency, is that it is never too late. There is no neuronal death. There is no problem of connections. There's nothing wrong. It's simply that Syngap1 is needed, and they are too low in Syngap1. In these studies by Gavin Ramba, what they saw is that you could get adult mice that have grown up without enough Syngap1, just like your family, your loved ones. And if you restore genetically, you give them back Syngap1 in the adult mice, they could rescue all the behavioral problems, memory problems, epilepsy, electrophysiology, so EEG. It's a disease that can be reversed, at least in part, even in adult individuals. So this makes Syngap1 an ideal disease to really try hard to get all of those gene therapies to get to trials, because in this one, it is never too late. And that's not the case for all diseases, but this is particularly good here. Also, the fact that you still have one good copy of the gene. So I want to summarize for you here, and this is a lot, and I hope it conveys that it's a lot of the opportunities that are there for developing treatments that could help in Syngap1. And these are all approaches for which there are things ongoing from other diseases. So this is not imaginations, these are realities. They just have to come to Syngap1. So at the left, I've placed the symptomatic treatment. The most obvious one are the treatments to reduce epileptic seizures, but we can think of treatments that will still improve all the symptoms. Usually behavior, cognition are the more tractable if they are to run those trials. But the important part is all the things at the right on the yellow part. So starting at the top as a clock going clockwise, we have all of these different modalities. We could think of increasing expression of Syngap1 using the DNA fragment, the ASO, like Stoke, like I was showing you, but Stoke is not the only company that could do that. And essentially you'll be acting on the good copy to have more, either by using the ASO, which is a small DNA fragment, or by using a normal drug, like Roche did for spinal muscular atrophy, that would do the same. They act doing the same. You just can do it with either approach. There are also the type of drugs I didn't mention that could have potential for Syngap1, and those are read-through molecules. A read-through is because in many rare diseases where mutations happen de novo, like in Syngap1, it doesn't come from parents, it just happens in the child. What happens is that the letter, the mutation, has created a signal that is stop, a premature stop in the middle of the gene. So the cell doesn't read through it, it just stops. It stops, and it can never produce the protein. So because this happens in many other diseases, there is a lot of research in developing drugs, and these are normal drugs, small molecule, the ones that you eat, that you take by mouth, that will allow the cell to read past that premature stop. They will always stop at the real stop because that's a very hard stop, but to read through. And they are so far not in trials in epilepsies, but they're being developed for other disorders. And I'm sure we will get to try some of this within the epilepsies, either on label or off label, if they work. And then there are several types of gene therapies, the one using virus, that should be able to work in Syngap1. One is the straightforward virus that carries the Syngap1 gene. The other is the one of the virus carrying the boss of Syngap1, the transcription factor gene. But then there's these newer approaches. They're still a little bit too early. We still haven't had that successfully done in trials, which are all of the different versions of CRISPR. I'm sure you've all heard about CRISPR-Cas9, all the things it's doing. That's the cut and paste of DNA. But what I'm thinking for Syngap1, more than the cut and paste, is the option where you use a virus to bring CRISPR to the brain. And what it does is to find the gene and activate it. And this is done with some of the variants. So this is similar to the boss of Syngap1. These are a lot of approaches. So I got to the end. I think I've spoken kind of fast, but anyway, we'll have time for discussions now. My summary for the opportunities for Syngap1 that I see is, remember, the rare epilepsies have revived the field of epilepsy. There is so much work, there's so much funding, there's so much progress, so much industry attention in our space that it is a good place to be, because we're going to be one by one solving all of these rare epilepsies. Second, you have a haploinsufficiency, the half sufficient. One gene is there, you have that half. Because of that, Syngap1 offers so many entry points to be able to design treatments to rescue. You could do it with a small DNA, with ASO. You could do it with a small molecule. You could do it with a virus. You could do it with a lot of things. And those treatments are not just symptomatic. These are non-symptomatic. These are the cures that we call, that are going to have the potential to treat all of the consequences of having reduced Syngap1. And that goes beyond epilepsy and includes communication, behavior, motor, all of the other neurodevelopmental aspects. Important point 4, Syngap1 is not degenerative, and the deficits appear to be reversible based on your studies in mice and what we know in the clinic that is not degenerative. So this is very meaningful. This means it's never too late. This means that we have a lot of patients out there, because there's so many still without diagnosis, that are going to benefit from these non-symptomatic gene targeting treatments. And the field, for all of the work that groups like you are doing, is really ready for clinical trials. You just need these trials to come and move in soon. And I want to highlight that, because you're my prime example of what I like to call the impatient patients. You've been building everything. You have that recent ICD-10 code, make sure it's in all medical classifications, the census, the natural history, a lot of the biology, the risking you've done, and a lot of the work of talking to every single company out there, make sure they hear about Syngap1, they hear about all of these resources, all of the work being done, and they give it a try. So I just hope it will pay off soon. And I mean, it will, it's just a matter of time, and you'll get all these treatments because you're making all the work. And yeah, prime example of impatient patients. So let's jump to the discussion. I hope you managed to follow. I spoke a lot. Marta, Mike, should I stop sharing? How we do?
I don't know what Mike is. Let's see, but this was fantastic. Thank you so much, Anna. I really appreciate it. Let's see. I'm gonna ask you a question that is kind of hard question, but I wanna know, you know, when we start treatments in medicine, like when they start doing all the genome sequencing, you know, the whole thing, they thought, I mean, it was gonna take, I guess, like 10 years and all that stuff. And how long do you think these treatments are gonna, what is the industry saying that it's gonna take to have all these treatments for these rare diseases, not just be a normal treatment, just like how we treat cancer or something else? Like, what is the industry thinking that this is gonna come everyday treatment?
Well, it's going to depend on the syndrome, because when they get approved specifically for a disease, for example, something that gets approved just for Syngap1, it gets there for Syngap1, doesn't get for the others. So some of these rare epilepsies are already having treatments approved. The first ones are still the normal epilepsy treatments, but some can be very, very good. Like in Draves syndrome, we already have fluorouracil that had seizure reduction to 70% and about a quarter of the patients seizure free. So big improvement from those. The ones that are designed to fix the gene or the protein are still in clinical trials. So it will be in a couple of years that we'll start having the first ones for Draves, but also, I don't know if you followed Marta recently for Angelman syndrome. Ultragenics had very impressive results also with one of those ASOs where they were treating teenagers, and some of the teenagers who had grown up nonverbal, because in Angelman that's a problem, began speaking soon after treatment. So you can do amazing things. Still not approved, still early stages. So we have, I don't know the number, it could be maybe 10 of the syndromes with clinical trials with this curative treatment. So in about 4 or 5 years, we're gonna have those with those treatments out there, and then the others will follow. I hope Syngap1 will follow soon after in the second generation. I don't think we'll get to all of them. We still have problems with the very ultra rare, and it won't be so standard to have treatments for those, but that is not your problem. You have a common enough rare disease. Yeah, couple of years for trials, couple more years for approvals.
Okay, and then everybody talks about the Syngap being a big protein and the big problem about being a big protein and maybe doing a viral therapy. And can you explain a little bit more in the sense that it's reversible versus not reversible and all those problems that we are going to have at some point have to talk about as a community?
Yeah. So when you're a company thinking of developing a gene therapy, there are many questions, not only the size. So ideally, we want the gene to be small because then it fits into the perfect virus, the super tiny one, the AAV. But in your case, if it's too large, you could always use another virus or do the boss of Syngap1. But there are many other things we look for. And you tick very well a lot of those boxes. So is it extremely, extremely rare, like 50 patients in the planet? Or is it rare, but there are going to be thousands and you're in the thousands? Good. That's good. That's going to be good enough. Do we know which cell needs the gene? Because the virus has to be programmed. Yes, you need the neurons. We know what it's doing. We know where it is good. We know where it has to go. Is it a disease where we can only act super, super, super early? Because after the damage is done, maybe they've lost some important neurons and then we can act? No, that's not the case. It's a disease where you're not losing neurons. There is no migration problem because maybe if the cells have gone to the wrong place, you cannot go back. There is no connection that is wrong. It's just that the protein is needed. And actually you can put it in older animals. At least we know that from mice and rescue. So that's very good. It means we can get all of the patients. We can get to those. So it ticks many of the boxes. The complex one is the technical feasibility to use the small virus. But if some companies are good at using large viruses. Some of the older viruses that were tried for gene therapy are lentivirus and are very used now for Parkinson and others. Adenovirus are in development also. There are all of these other approaches where you have the boss of Syngap, a transcription factor, or you use the CRISPR version to activate the gene. So you have adenovirus, you have many options. And you tick the important boxes. Do we know when we need it? Anytime. Do we know where we need it? Yes, in the neurons. Yeah.
And then another question always come up with Syngap or with viral therapy is the distribution on the brain, because of course the recent studies are diseases more that are in the spine, and it's easy to get there because, I mean, it's so small, but the brain distribution, and we think now the big problems are happening the midbrain, like hippocampus and all the stuff that is harder to get into.
Yeah, that is a problem for the entire field of neurology. Yes, when you have a focal neurological disease where only a little bit of the brain is affected, you could inject the virus there and it will cover. When you need it everywhere in the brain, like Syngap, but like all of the epilepsies, even the best virus, the AAV, might not be enough. And there are a lot of things ongoing to try to address that. There are companies developing modifications of AAV to get one that will really cover a lot. There are others just doing different route of administration, even thinking of using vagal nerve and different routes. There are companies doing naked gene therapy. Forget the virus. And that's like Generation Bio and some of the big ones. They're doing DNA or even RNA. This is like the vaccine from Pfizer and Moderna. Just doing straightforward DNA or RNA cover with a little ball of fat. So there are so many options that you will be able to benefit from those. That's a common problem, and that's an important problem. There are companies working on getting the virus or the DNA or the protein from plasma to the brain through the blood-brain barrier. And they're working for that for neurological diseases. So you could use the blood system that goes through your brain, and it's like every neuron is just 2 cells away from a blood vessel, and get it to then cross. There's so much ongoing. It's kind of crazy to follow, but there are many, because for a lot of diseases, notably for Alzheimer, where there is a lot of work, we really want to be able to cover all of the brain. So that will be addressed. There are a lot of options there.
And now that you mentioned about the vaccine, one of the questions that we get now when the vaccines are getting approved now for teenagers and then for children is there was a question several times, like if there is a contraindication of getting vaccines just because you are thinking about getting some viral therapy later or some.
Okay, I thought you were going to ask more, because some ask people with epilepsy, the vaccine could cause some reaction and then make them worse. You're really thinking of the virus. Yes, that is a very good, that's a very good question. That's a very good question, because a problem we currently have with gene therapy is that you cannot use it twice, because the first time that you give a person the virus with the therapeutic gene, the body is going to recognize the virus, and just like a vaccine is going to create immunity against it. So the next time that you try to give them the same virus, they will kill it. And that's how it works to defend you from infections. But what if the first time that virus was a COVID vaccine, and now you're ready to kill it, and the next time it is a gene therapy for Syngap? That is the question. That should not happen with the COVID vaccines for 2 reasons. One is this is a different virus. They're not using the type of virus that we use for gene therapy. But the main one is they're just using a teeny bit amount in the muscle, and it's not comparable to what we're going to need. So that will not immunize you against that virus, even if we were to use the same, which is the reason why you can have a second shot for the vaccine and a booster. You're still not killing the virus. It's too little what we're given. So the viruses are different. The virus used for the COVID vaccines are not the same ones used for gene therapy. They won't create immunity for the gene therapy. And in the odd chance that someone were to do the COVID with an AAV9, because it's just a little bit in the muscle, it's not enough to create that, which is why we can have several doses. Yeah.
And there is any preference on the type of vaccine? I mean, I have here maybe the Pfizer, just because the type of virus and the type of vaccine it is, it's a little bit preferable. But I don't know if that really has any.
As compared to the other viruses, I don't know there. Yeah, preferable for what? Then there is whether the side effects will be harder for some kids. It seems that at least, I'm trying to think, at least AstraZeneca, some of those have like stronger side effects. Even Moderna seems to have stronger side effect. If patients with Syngap1 and people with many neurodevelopmental diseases, they tend to be a bit more fragile. So the milder side effect, the better, just because you don't want to get them sick. But it's not something tremendously specific. It's not like putting them in danger. It's just like trying to avoid from getting them exposed to the flu, and yeah, because of the more overall fragility.
Okay, let me see.
It's still very recommended to vaccinate any person that is medically fragile for COVID, for sure, because that is the one that can cause a lot of harm.
I mean, yeah, I tend to think that's the right now problem that we have. If you get, in my case, if you get a teenager with Syngap and has to be admitted, I don't know how we manage that. Then, yeah. I think that's one thing that is more close than having the other problem that we are trying to get the cure for Syngap is close by or something. Let me see if we have any more questions in the, I think
Peter had the same question.
Peter has the same question. I think I don't see any more questions right now.
If you have it live on Facebook, people could be asking on the Facebook feed, and you wouldn't see that here on the webinar.
Oh, I don't know how to open that one. I have to ask Peter.
Because in other webinars we've done for the diseases is on Facebook where the activity happens, and you could miss it here.
Then as a community, I mean, you kind of know us a little bit or kind of well as a community. Anything that you recommend besides everything we have done that we are trying to get is, I mean, natural history together, anything that as a community you would recommend? No,
I think you really, 4 years ago or so, I thought I was leaving the rare disease Spain. So I wrote this is space, not Spain. I wrote like a manual, this impatient patients, and you seem to have followed everything I thought were the right things. You're really doing a great job on that. The main thing I encourage is to have conversations with pharma companies, to biotechs, understand their thinking, how they see the disease and what they see as the main issue. One aspect of Syngap1 that I find from my pharma brain that would be very attractive to me is the knowledge that there is a lot of people out there with Syngap1. And if you can try to get those numbers, and until you get the numbers, at least kind of white paper articles, the message out there, like, hey, this is a hugely underappreciated disease. If we were doing genetic testing to kids that might not have so much epilepsy, or maybe not at all, you will realize how many people with intellectual disability have Syngap1. Uncovering that number is what will get you to the top of the list of rare diseases, because they often look at that. What is the most common of the epilepsies? What is the most common of the DEs, of the neurodevelopmental? And you actually have a much more common rare disease, but it's poorly documented. So in addition to understanding from all the companies what they see and exactly where they pay attention and what convinces them, I think that one, which is a clear selling point to get them interested in Syngap, is like, hey, we're talking about one of the largest of all of these rare neurodevelopmental diseases. To get that, if not the number, at least the message that it's going to be much larger than you think. Stay tuned, we'll come up with it, but that is what will get them. Yeah,
but genetic diagnosis is such a battle everywhere. I mean,
yeah,
it's, that's the difficulty for us, and I guess for many countries to get there. But like you said,
and for epilepsy, we're doing it more, and it's a little bit easier when you have a young child and they start having seizures. And if the first drugs also don't work, then usually they might get tested, and it depends on the country. I know, at least in the US, there is a free genetic panel at Invita for all children 8 and younger that have at least one seizure. So hopefully that gets more and more known, and more families get sent that route. But in the case of Syngap1, it would help that any child that has some signs of neurodevelopmental delay will get through one of the panels, because at that time is where you will uncover a lot of them. But that's not yet there. For seizures, yes. For neurodevelopmental, no. Potentially because there are treatments for seizures, for epilepsy, more than for the others. It'll get there.
Yeah.
Let's see, Peter. So, no, okay, good question, Peter. It's a kind of ambiguous answer. If you receive an ASO, would that disqualify you then from getting other modality like an AAV? If it is approved, so the disqualification is, it is not possible for a person in any disease to be at the same time on 2 experimental medicines. So you cannot be on 2 clinical trials, and you cannot be taking an experimental medicine and be in a clinical trial. By experimental, I mean, maybe it's not a trial, but it's like the cannabidiol oil produced more from how you call that, dispensaries and that. So that's the problem. So if you are on an ASO and it gets approved, you can keep taking it, just like you can take clobazam and any drug and get on a trial for an AAV. But until it is approved, you couldn't be taking both if both are experimental drugs. That is the regulatory reason. It's not compatible to have two experimental drugs. Now, let me tell you the financial reason. In spinal muscular atrophy, there are 3 treatments that can fix the gene and help the children. One is an ASO, that's Spinraza. Then there's the gene therapy by Novartis, that's the virus, that's the 2 million a year famous medicine by Novartis. And then there is the small molecule by Biogen. You could technically give all 3 to the same child. The reason it's not done is financial. You don't want to be paying a 400,000 a year drug plus a 2 million ones plus. So there will be reimbursement issues that might make clinicians have to choose if they want to give that child an ASO or an AIV, because both are 2 very expensive treatments. You could always combine with the small molecules, with the normal drugs, those are cheaper. So medically and regulatory, no problem if they're both approved. Reimbursement, it's not working, at least for SMAs, too much money.
That was a very good question. I haven't thought about that one.
And now there are other, like I work a lot, half of my time in CDKL5 deficiency. There are 3 gene therapies getting close to clinical trials. What the families are asking is, how are we going to choose? So suddenly there's going to be 3 clinical trials, and these are virus gene therapies. You can only have one in your life. How you choose, and what if you sign up for one and it's a dose that is too low, or it doesn't work so much, and the other is the good program? And yeah, that's a good problem to have, too many, but it's indeed not a bad question.
It's a big, yeah, it's a problem, because if you have only one chance, and that's the problem with those therapies. That
yeah. Well, the good thing of ASOs is that because you have to keep redosing, and every few months there is another shot. If it's not working, you just stop, and it's like a normal drug, and then you're clean, and then you can get into the AAV, for example. And it's just that you cannot be on the trial at the same time taking both, because regulatory decision is like, now we don't know if your improvement or your side effect is one or the other. It has to be clean. As long as one is approved, you can then be on the trial for the other. If both are approved, then it's too expensive to be on both. Yeah.
And where are do you see these trials? I mean, I guess in the US is the easy place to run these trials, although a lot of medicines now are getting done outside the US because it's easy to set up trials. And
Depends.
I guess the European Union sometimes also is difficult to work through because the regulations then.
Yeah, but here's the good side of Brexit. So the problem we had in Europe is that you needed 28 countries to agree, because the EMA has representation of all of the countries. But now that the UK is independent, they have a very good regulatory agency called MHRA, and they were always leading a lot of the thought leadership here in Europe for gene therapies and advanced therapies. So not traditional drugs, but all of the cool biotechnology treatments. And now that they're separate, they are making it very easy for companies to run gene therapy and ASO trials in the UK. So US and UK are the obvious countries to run the first trials, because the agencies are very knowledgeable and they help a lot, while old Europe with the 27 or however many we have, it's still a lot more bureaucratic, and it's going to take longer for trials. Usually for the first studies, phase 2, which are smaller, UK, US. Afterwards, when you need to do global, and yes, then you get Australia and Canada and Europe. For South America and others, yeah, I don't know, probably wait for approval and then see if you can access. Yeah.
Okay. Let's see. I think no more questions. I don't know if somebody else have any questions, but this was great, Anna. I think it's always so informative, and so nice to see how everything is getting developed. And I think you have to show to the families how clear, I mean, very in a clear way, what possible therapies we are gonna see in the future. Then thank you so much.
Thank you.
I appreciate this talk. I think it's gonna be one of those talks that every family is gonna go and see, because we will put this in our YouTube channel. And I think this is a talk that every family that is new wanna see and get informed where we are with therapies. Then I appreciate it very much. Thank you for this.
Thank you. Thanks, everyone. Bye.
Thank you. Bye-bye.